Hi, let's continue to study about intestines. So there are small and large intestines, which is for absorbing water and nutrients. And also we'll discuss about fluid balance in our body. So first, let's discuss the layered structure of intestinal tube. Before we talk about it, we have to think the structure of our body, especially inside the abdominal cavity. So abdominal cavity looks like this. So there exists a peritoneum, and that's lining of this blue. And interestingly, our internal digestive organs, such as stomach, liver, pancreas, and intestines are something like this balloon. It goes inside this peritoneum. So it looks like this invagination of this visceral organ. So that structure looks like this. And if this is big, it looks like this. And that this blue lining is called serous membrane. Okay, so let's look at our intestinal tube. Because of this, you see these two linings are attached together. So that contains the structure of this intestine and its linings. So there are a few anatomical structure which I want to discuss with you. So first, because intestinal tube inside this hollow place, space is called the lumen. And second, and this blue lining, which is called a peritoneum, is a serous membrane that forming the lining of abdominal cavity here. So this blue lining is called peritoneum. So sometimes when you are giving drug or anesthesia to mouse into the abdomen, this is called intraperitoneal injection, which means you have a syringe, you poke through this peritoneum and then you inject. And that fluid will be absorbed slowly and getting into your circulation eventually. And the second is called a mesentery. If you took out of this gut, let's say during experiment, you will find that there's a thin transparent layer that composes of blood vessels inside. And that is called the mesentery. This is, you can see from this structure, and that lining put together is forming this mesentery. And mesentery is a fold of peritoneum which attaches to stomach, intestine, and this any organs inside, and the other part is inside the abdomen. So that's called the mesentery. And let's look at the inside the lumen, inside the intestine. So the intestine inside is covered, as I said, a mucus. So there's the outermost is called the mucous membrane. And right underneath this mucous membrane, the connective tissue we call lamina propria. And the finer part is connected to muscular. So that's called the muscularis mucosa. These three layers put together we call as mucosa or mucous membrane. This is a membrane that lines various cavities in the body and covers the surface of internal organ, and we call it as a mucosa. Of course, underneath the mucosa, there is a submucosa, which is also connective tissue that touches to outer muscular layer. So that is called submucosa because it's below the mucosa and connects to the muscular layer outside. So these two layers of this muscles are composed of muscles, and outer side is called a serosa or serous membrane. So this is smooth tissue membrane consisting of two layers, inner and outer layers. And so you can see these muscles, interestingly, there's two layer structure, inner and outer, we call muscularis externum. So how does that actually prepare food while performing its digestive function? And look at this. This is mouth and down here is the towards rectum. Then let's see. So first you have to have food intake, this food. You have to grab and squeeze top part, and then needs a circumferential action of this muscle, while this part has to relax so that it can push, prepare the food. And so I have to define two terms called proximal and distal. So in the body, more of a trunk or central part is called, we have called proximal, while this outer side is called distal. So in this intestine, inside part is a proximal, while towards the anus, because it expels food, the digestive stuff to outside. So that's we call distal. So the definition is important, and to push from the proximal and to distal. So you also have to, along the longitudinal direction, you have to also muscles to push like this towards the distal part. So think about grabbing food by squeezing the circumferential, and at the same time moving the material, the downsides, you have to also push outside. So that's a longitudinal. So this two muscle layer, inner and outer layer, inner is circumferential or circular muscle, outer layer is longitudinal muscle. So these are smooth muscle cells, one along the intestinal axis and the other circumferentially for mixing content and propulsion along the intestine. This is very interesting structure. And the rhythmic contraction happens quite slowly. You can see stomach for 1 minute, about up to 3 times of this squeezing happens. And in small intestine, the upper part, the duodenum, it takes about 8 to 10 times per minute. So it's like every 6-7 seconds, it will like squeeze. Okay, and these propelling movement is called, we call peristalsis. Okay, that's a muscular event that propels food through the intestine, and that is a coordinated contractions of distal part is released and proximal part is constrict. So this way food can propel and through this peristaltic movement. This is a reference. So in biochemical point of view of a nutrition absorption, and you can see this small intestine part, depending on the size of the arrow, a lot of carbohydrates, protein and lipids are absorbed in the intestines, small intestine, and here bile acids. And at the more cellular level, you go down into epithelium, which is the outer lining of the intestine, which directly touches with this food element. So let's see how the digestion happens. So on example, glucose is directly passing through the epithelium. So intestinal interstitial space, which is going towards eventually to hepatic portal vein as glucose directly goes, which increases our glucose level. And protein will be decomposed into amino acids, and amino acids can be uptake through this epithelium. And what about sugar? Sucrose, or like this, sucrose will be decomposed to glucose and fructose, and they can pass through this epithelium. And also peptide, proteins component. This peptide can be hydrolyzed and decomposed into single amino acids, so which will form our nucleic acid and a protein. And also fatty substance, triglyceride, can decompose into glycerol and fatty acid. They actually are synthesized inside as a triglyceride. So now think about the fluid balance in the body. In fact, there's a lot of fluid getting into our lumen, and we reabsorb most of the part. So let's see the balance. So this is just an example, but one day in the body, the fluid inside the food is about 1 liter, okay? And the fluid intake is water or drinking, and we intake about 1 liter. And interestingly, our saliva, which is produced almost 1.5 liter a day, so it keeps coming out. And also gastric juice inside our stomach, gastric juice is even 2 liters of juices are produced inside our stomach. And how about pancreas? Wow, pancreatic juice is 1.5 liters secreted into our duodenum space, okay? And also when you talk about liver, there's a bile fluid. That bile fluid secreted is also 0.5 liter. And our intestinal fluid, through the intestine, there are fluid secreted, is almost 1 liter. And that adds up about 8.5 liters of fluid are getting thrown into our intestinal tract. While how much do we reabsorb it? Let's see. Small intestine absorb about 6.5 liter a day. Large intestine, almost close to 2 liter a day. And also our stool contains its water, and that's about 100 milliliter. That adds up 8.5 liter. So in a nut, we have a fluid balance in our body to keep our homeostasis or ourself at a steady state. So digestive system secretes large volumes of fluid, which we call digestive juice, to facilitate this digestion. And most of the fluid you can see over here is reabsorbed in our intestines. Only those water in stool, a little amount will be coming out of our body. So I just want to show you the structural differences between large and small intestine. Small intestine has a lot of this villi coming out, while large intestine is on the opposite way. It's called the pit. So both are increasing its surface area, but they look a little different. And I just want to give you an example of that very interesting architecture to effectively increase surface area in the small intestine. Think about when we talk about this respiratory system, we are adopting the small alveoli, which is only 300 micron, and that's small little ones, but we have 300 millions of them to increase its surface area almost 80 square meter, which is almost the half of the tennis court. So that big space, we are using it for gas exchange. Now for nutritional absorption, we are using a little different strategy, but also effectively increasing the surface area. And that shows like this. We have small intestine. We have, you see this circumferential fold, which effectively increased the surface area by about 3-fold. And when you look at inside more closely, you see this like this villi structure. And also this fold contains outermost mucosa and underneath submucosa, which connects to the muscular layer called the muscularias. And each of these fold, you can see this little protrusion, which is called a villus or multiple villi. You can see this villus is covered with epithelial cells. And this epithelial cell underneath, there's lots of capillaries and lymphatic. Specialized here is called lacteal in the intestine. And that forms this shape of villus, okay? And multiple villi. And of course, it's connected to the vein, artery, and lymphatic duct. And if you look at this single epithelial cell, and that single epithelial cell, this apical part is not smooth. It has, again, a lot of this fine like finger-like structure that's called microvilli, which effectively increase the surface area by 20-fold. So let me give you a table to compare the small and large intestine. While small intestine, because it has smaller diameter, 2.3 to 3 centimeter, versus large intestine has 3 to 4, 5 centimeter diameter. While small intestine is longer, like 3 to 6 meters, while large intestine is about 1.5 meters. Small intestines helps digestion and absorption of nutrients. Large intestine also helps reabsorption of the remnant food and also elimination of waste because it forms a stool. In terms of absorption, small intestine absorbs carbohydrates, proteins, fat, minerals, and vitamins, while large intestine absorbs water and some nutrients and salts. And small intestine is composed of duodenum, jejunum, and ileum. And that's connected to the cecum in the large intestine. The spelling is it's English, but the American English has a cecum, C-E-C-U-M, plus colon. The colon composes of ascending, transverse, and descending colon. And that connects to the rectum and also anal canal. So let's review about this small and large intestine. Small intestine is thus the longest section of GI tract or gastrointestinal tract, which absorbs most of the nutrients. And also it's a high volume water absorber because it's almost 5 to 7 liters per day. And the surface area, as I told, the circumferential fold gives about 3 times increase, and the villus structure increases about 10-fold. And up to the epithelium, single epithelium cells has a fine microvilli we sometimes call brush border from pathology point of view because that looks like hazy brush from light microscopy. And that even increases 24, which results about 600-fold increase in the surface area relative to plain tube. So this is a strategy that our small intestines is adopting. And that architecture is about 6 meters long and about several centimeter diameter and 3-fold circular folds, 10-fold of villi, 20-fold microvilli. And very importantly, this is consistently meeting with outside food product or sometimes toxins and harmful substances so we can intake. And so it has a relatively rapid turnover in this epithelium, which lines villi. You know how often do they actually change or fluff off? It's every 3 days. And because of this becomes also a problem that it's also susceptible to insert if that influence mitosis because it keeps happening. For example, a cancer patient who have to undergo radiation therapy, which damages the DNA, so it affects, it kills mitotic cell. Then small intestine cells are keep generating, and this radiation exposure makes the small intestines, it rapid turnover, and it kills these cells for temporarily. Then as a side effect, the patients can go to diarrhea. Okay, so that can become a issue. And in comparison, large intestine, you can say as a drying organ because it makes a finer stool. So it absorbs remaining water 1 to 1.5 liters per day. And also it stores undigested wastewater and material and allows it to release under our voluntary control when we want to go to the restroom. And also a very interesting part of large intestine is it harbors a bacteria, which is symbiotic to ourselves. And that has a specific role, which is still under very hot issue these days. And that bacteria converts dietary carbohydrates to monosaccharides for our absorption. So with this, we discussed about digestive system. Eventually, what it does is breaking down of food into molecular components, and then nutrients will be being absorbed into our intestinal system and into the body, and liver also stores some of it as a glycogen. And for that, these interesting structure of layer structure in intestinal tubes, and our stomach and pancreas, liver, and small and large intestine has been covered. Thank you for your attention.